About the effectiveness of the bioresonance, people always say a lot, lots of various information. Someone says that he doesn't believe in it. Someone, on the contrary, believes that this is the medicine and the technology of the future. Actually, that's what I think. Because bioresonance is those technologies which can allow a huge number of people to regain their own health, which can prevent people from getting sick, and to be free in their movement, in their life, and most importantly, in their own health and their loved ones. That's why, probably, I have devoted so many years to this. And now we are sweeping into those areas which may not be related to the human being directly. Our company pays a great attention to the various scientific tests and examinations. And in recent years, I'm basically experimenting only with in vitro mode. This is interesting for me because such a mode is out of the human being, under the microscope, denies any arguments. About the placebo effect, I believe or I don't believe, works or doesn't work, which means we shoot everything under the microscope and it's good. But the complexity of working with a microscope lies in the fact that many of our infections have a very small size, and accordingly, shooting such a beautiful visual video, it is sometimes very difficult. So we decided to make it a little bit in a different format. Because we already carried out tests directly on infections that are related to the human being, we carried out tests on pets. We conducted tests on domestic insects, if I can say so. I'm talking specifically about bees. But now we decided to conduct tests on crops. In fact, this is a little bit an audacious experiment, I'll tell you right now, because I didn't have any relations with the agricultural field. In principle, I didn't know that in this field exist real serious problems. I didn't know that such a problem as potato nematodes or strawberry nematodes bring huge damage to the agriculture. And we decided to aim this particular area of agriculture, because we found out what turned out to be those means which would allow to 100% to deal with any nematodes in the ground. Simply doesn't exist. It turned out that the biggest effect that can get in the treatment of those same potato nematodes is up to 18%. And there are no chemical substances, which would give a higher percentage. It really surprised me a lot, and I decided to try it with my colleagues. Why wouldn't we try our devices to make an influence on such pests? And to my great joy, and to the surprise of my colleagues, these experiments went great, because even at the first connection, and not yet well-functioning programs, we already got efficiency around 50 to 70%. It is clear that if we speak in the future about some technical embodiment of recovery for agricultural products, you need to conduct fairly decent tests. Selection of radiation power, radiation time. But this is a completely different story. This will be our future development. Even the initial tests, they turned out to be just fine. For the tests, we invited doctors of biological sciences, biochemistry, parasitology, and medical genetics, Milishnikov Alexander Nikolaevich, who kindly agreed to help us with the organization of research activities, with the search and the culture extraction, as well as the description and systematization of the obtained results, acting as a scientific consultant and an active participant in the experiment. All my life, I was engaged in basic research after graduation. At the very beginning, I had to deal with cotton. I was engaged in wheat and in caracol sheep. Most of the time, we were engaged in population-based study, so there is animal population genetics. But I have always been interested in medical problems. In particular, health improvement, body cleansing. And I was invited to study pest agricultural crops, nematodes, because in practical terms, it is a very important problem on a global scale. Actually, and not only in our country. New methods of chemical exposure are being developed, but there are pests that cannot be destroyed in such a way. Electromagnetic influence technologies are being developed in the company Data Ellis on biological objects. In particular, appeared the topic of the use of the bioresonance electromagnetic fields on agricultural pests. What we actually did. Like the human nervous system, an electrical activity is present in the nervous system of nematodes, which indicates the presence of electromagnetic fields with different intensities and frequency ranges. The objects of the study were two different species of nematodes. The first species included phytoparasitic stem of the potato nematode Didalenchus destructor. This type of nematode is a real problem in agriculture. Annually destroys a significant percentage of the crop. Parasitic sample preparation of the potato stem nematodes carried out on the day before the experiment directly from the affected potato tuber. The nematode suspension was transferred to the middle of the plastic petri dish measuring 3.5 centimeters. The electromagnetic impact on the experienced nematode samples in petri dishes was made using the portable device DeVita AP, manufactured by Data Ellis Holding. Before the beginning of the experiment, it is noted that most specimens in control and test plates were fixed. Neither in the control nor in the test samples, no individuals with any damage were noted. 1 hour later, in an hour of working program, we got a clear picture about the effectiveness of the device DeVita AP. Multiple biological breaks in the shells of nematodes, their paralysis, and even defragmentation. By this time, among the individuals began to be noticed a damage in the form of vacuolization of the internal space, as well as a massive cuticle cuts among the females, with access to the media content. There was a noticeable increase in viscosity of the medium due to being hit by the protein mass of the broken nematodes. In case of an electromagnetic field exposure on biological objects, the main task is to consistently generate signals on frequencies with high accuracy corresponding to those produced by pathogenic agents or parasites in the life process. By entering into resonance with the biological radiation of their organs and systems, we cause bounce, overload, and subsequently the structure destruction of the influence object. In the case of the correct sequence, selection, and exposure time, we get a destructive effect, which is clearly visible on these frames. Due to the flexibility and the relative simplicity of the procedure adjustment of the software, which changes the numerical code and the frequency sequences, we can achieve unsurpassed speed during work, adaptation of the program, and increase of the effectiveness impact, quickly making adjustments in accordance with the observations obtained during the experiments. The approach, in the case of a parasitic potato nematode, allowed us to achieve 50% efficiency in vitro mode for several samples, while the most productive for the moment is the chemical solutions for this problem. Known to us, they cannot overpass the efficiency limit of 18%. To the second type of nematode studied by us belongs to the free-living vinegar nematode. The suspension of vinegar nematodes prepared from uterine culture, supported on a substrate from chopped carrots mixed with breadcrumbs. Unlike potato nematodes motionless at rest period, vinegar nematodes have a high activity. When using the exposure scheme with repetitive high frequencies, a relatively quick mortal effect was obtained. The exposure duration on the test samples was also 1 hour 30 minutes. As a result, already in the first minutes after the beginning of training, we observed 2 to 3 seconds cramps followed by the activity restoration. The massive drop in activity and deceleration of nematodes was observed at 40-minute exposure to the electromagnetic field. An hour later, we saw the same familiar picture with breaks and the end of the internal contents of the nematodes on the medium. The initial entering signs into the resonant interaction occurrence of the biological field of nematodes of the electromagnetic field of an external source expressed in the fading of the movement of the nematodes for a few seconds. Convulsions, transverse and longitudinal projections along the entire length of the individual twisting, individual nematodes into nodes and collecting into tangles. Paralysis of representatives of the studied species under the influence of electromagnetic radiation began at the tail end part. In the last part of paralysis, seized nematodes with a nervous knot. Meanwhile, the large old females are practically died. Part of young females, as well as males and larvae, remained motionless. The aim of the experiment was to identify the effect of the electromagnetic radiation with different structure and intensity on nematodes' viability in the concept of the electromagnetic radiation. Includes parameters such as direction of frequency, change from high to low, and on the contrary, repeating small frequency ranges several times with different periodicity, time of exposure, radiation, and so on. It's namely from a competent selection of a sequence, frequency, and accurately calibrated the time of exposure, and depends on the efficiency of the bioresonance effects. In the case of vinegar nematode, the effectiveness of our experiments in the observed droplet suspension reached a score of 80% in vitro mode. Besides the fact that this method proved itself to be excellent at the beginning of the experiment with direct irradiation of culture, the effect after the execution of the program we could observe even after 1 day after the exposure. When I was invited to conduct these experiments, I absolutely didn't believe that it is possible with low-intensity emissions to obtain some real results. But the very first experiments showed that there was an effect. When nematode cultures are exposed to a bioresonance field, first they are immobilized, and then they begin to die, burst, and at the end, there is a complete death. These are the first results. They are very encouraging, which show, firstly, the effectiveness, and secondly, it allows us to enter into the practice of combating agricultural pests. Test in vitro, and namely, and on agricultural pests, will remove from a huge number of people such questions as works or does not work, effective or ineffective, because on these little basic nematodes, which a person can't see, but under a microscope, they turned out to be very noticeable, very visual, and we actually saw in real time how they collapse under the influence of the electromagnetic fields directly. I think this is our future, a future breakthrough, and I think it is, of course, a patent and an invention, and many other scientific works that we will do in the future.